Hello everyone, today our group will be presenting about paper chromatography in pharmaceutical analysis. First, let me briefly explain what chromatography is. Chromatography is a technique used to separate the components or solutes of a mixture based on the relative amount of its solutes distributed between a moving fluid stream, known as the mobile phase and a stationary phase. According to Keeler and Roy in 2026, this principle allows different substances in a mixture to be separated effectively. Paper chromatography is widely used in pharmaceutical analysis for several reasons. Firstly, it provides precise and sensitive analysis by allowing researchers to evaluate the purity, stability, and consistency of drug throughout the development, manufacturing, and quality control stage. Secondly, according to Ali Gamal Al-Kaf in 2024, this technique is an excellent tool for analyzing bioactive compounds present in pharmaceutical preparation and medicinal substances. Furthermore, chromatographic technique enables both qualitative and quantitative analysis and allows substances to be compared based on their compositions. There are three objectives of this study. The first objective is to determine the purity and stability of drug substances and formulation for quality control purposes. The second objective is to identify substances through chromatography separations for training, counterfeit detection, and low-cost drug testing purposes. The third objective is to monitor reaction process and verify the accuracy of colorants used in pharmaceutical formulations.
As shown on the slide, chromatography is a technique used to separate components in a mixture. This separation occurs because different substances have different mobilities towards the stationary phase and the mobile phase. Specifically on this slide, the stationary phase is the cellulose, the mobile phase is the solvent that moves up the paper by capillary action. Looking at the diagrams on the right side of the slide, we can see that this process in action. It starts with the sample mixture placed on the baseline as the mobile phase travels up past the sample spot. The components are separated into distinct levels based on their mobilities before the mobile phase reaches its maximum front. Next, we look at how paper chromatography works in pharmaceutical analysis, specifically its working principle in the pharmaceutical industry, based on SOPHE ETAL 2026. According to the slide, the process follows 5 main steps. First, the sample is coated on the chromatography paper. Second, the paper is placed with a solvent chamber. Third, the solvent moves upward and carries the compound. Fourth, different compounds travel at different speeds. And the last one, the separated spots are identified using the RF blending. Moving on to the application in pharmaceutical analysis. As listed on the slide, there are 3 main applications. First, the detection of impurities in drugs. Second, the quality control of pharmaceutical products. And third, the identification of active pharmaceutical ingredients. That concludes the application for this part. I will pass the presentation to the next speaker.
Now, I'm going to explain the methodology, but before I proceed, these are the materials that we need to do the experiment. Next is the methodology. The first step is sample preparation. A pharmaceutical tablet such as paracetamol or aspirin is crushed into a fine powder using a mortar and pestle to increase its surface area. A small amount of the powder is then transferred into a test tube and 2 or 3 ml of ethanol is added. The mixture is shaken to dissolve the active ingredient and then filtered to remove insoluble excipients, producing a clear sample extract. Standard solution of pure paracetamol and aspirin are also prepared separately in ethanol for comparison. The next step is preparation of the mobile phase. A solvent mixture of ethyl acetate and ethanol is prepared in the required ratio and poured into a chromatography chamber to a depth about 0.5 cm. The chamber is covered and left for approximately 15 minutes to allow saturation with solvent vapor, ensuring better and more consistent chromatographic separation. The next step is applying the samples onto the chromatography paper. First, a baseline is drawn about 1.5 cm from the bottom edge of the chromatography paper using a pencil. 3 evenly spaced marks are then made along the baseline using a capillary tube. A small spot of paracetamol standard is placed on the first spot, the aspirin standard on the second mop, and the tablet extract on the third mop. Each spot is allowed to dry before adding another drop if needed. Small and concentrated spot helps achieve better separation. Next is the development of the chromatograph. The chromatography paper is placed vertically into the developing chamber, making sure that the solvent level remains below the baseline. The chamber is then covered, allowing the solvent to move upward through the paper by capillary action. As the solvent travels, the sample components separate based on their different affinities for the paper and solvent. When the solvent front reaches about 1 cm from the top of the paper is removed. The solvent front is marked immediately with a pencil, and the chromatogram is left to air dry. Finally, the chromatogram is analyzed. After the paper has dried, the separate spots are observed and compared with the paracetamol and aspirin standards. The RF value of the spots can be calculated to identify the active ingredient present in the pharmaceutical sample.
To confirm the identity of the compound, we calculate its RF value. RF stands for retention factor. The formula is RF equals the distance traveled by the compound divided by the distance traveled by the solvent front. In this example, the solvent front traveled 8 centimeters and the paracetamol spot traveled 5.2 centimeters. The calculation is 5.2 divided by 8 centimeters, which gives an RF value of 0.65. The RF value of the pharmaceutical tablet extract is then compared with the RF value of the standard solution. If the values are the same or very similar, it indicates that the unknown sample contains the same compound. In this example, the tablet extract has the same RF value as the paracetamol standard, confirming that the tablet contains paracetamol. To confirm the identity of the compound, we calculate its RF value. RF stands for retention factor. The formula is RF equals the distance traveled by the compound divided by the distance traveled by the solvent front. In this example, the solvent front traveled 8 centimeters and the paracetamol spot traveled 5.2 centimeters. The calculation is 5.2 divided by 8 centimeters, which gives an RF value of 0.65. The RF value of the pharmaceutical tablet extract is then compared with the RF value of the standard solution. If the values are the same or very similar, it indicates that the unknown sample contains the same compound. In this example, the tablet extract has the same RF value as the paracetamol standard, confirming that the tablet contains paracetamol.
Okay, now we are talking about advantage and disadvantage of chromatography in pharmaceutical analysis. For advantage, we have high sensitivity and precision, where the chromatography technique actually can detect and quantify an active pharmaceutical ingredient, and the impurities even is at extremely low concentration. Next, chromatography has an excellent separation capability to effectively separate a highly complex mixture for assessing drug purity. It is also versatility, where it is capable to a wide range of molecules, from small molecule synthetic drug to large biomolecule like protein and monoclonal antibodies. A modern chromatography system are also fully automated with auto samplers and advanced software that allows for continuous, unintended analysis of large batches of samples in quality control laboratories. And chromatography methods like HPLC and GC are universally accepted and standard requirement by regulatory body for drug approval. Last but not least, chromatography are also hyphenation, where coupling chromatography separation with mass spectrometry allows for simultaneously separation, quantification, and structure and identification of unknown impurity or drug metabolism. For disadvantage, these analyses are usually having a high operational and equipment cost, where the instruments require a significant initial capital investment and maintenance. Mostly, chromatography are destructive to the sample during the detection process, where the specific sample injection cannot be recovered for further biological testing. It is also require time-consuming for sample preparation, which can introduce to human error. In addition, techniques like conventional HPLC consume large volume of organic solvents, that leading to a high chemical waste, disposal cost, and environmental concerns. Furthermore, chromatography techniques are complexity and required for skilled personnel in analytical chemistry. Lastly, unlike some spectroscopic methods, chromatography needs a real-time inline monitoring directly inside a manufacturing vessel without a sampling delay. In conclusion, this analysis successfully achieved all its core objectives, proving that chromatography is a definitive tool for ensuring pharmaceutical quality and safety. The application fully verified and the purity and stability of drug substance to satisfy strict quality control standard and successfully identify chemical substance to validate its use in training, low-cost testing, and counterfeit detection. Furthermore, the analysis successfully achieved accurate process monitoring by tracking chemical reaction progress and verifying the exact composition of colorant in final formulation. Ultimately, the complete fulfillment of this goal highlights chromatography's indispensable role in maintaining manufacturing integrity and safeguarding public health.